Hello friends, welcome to Meditation 101. Today we'll talk about reversible cell injuries. Okay, so I do have uploaded on video based on the cell injuries or whatever the causes are there or etiologies, you can just watch out in my channel, okay, in the cell injury pathologies. Now, before starting the topic, so let's go to with the very basic things. So what is the most common cause of the cell injury? So if I write over here the most common cause, MCC, most common cause, okay, of cell injury, definitely that is the hypoxia, that all you know it, okay, that is the hypoxia. So hypoxia means what? We know that whenever there will be deficiency of oxygen, okay, that will lead to the formation of hypoxia. Now it's very easy to understand that where oxygen will be utilized. Now we know from the very basic things from our school knowledge that we do have some steps in the aerobic respirations from where we just get the ATP molecules. Okay, so if we just talk about all these things over here, we know that the glycolysis. Okay, we know that TCA cycles, so the Krebs cycles, and we know that electron transport chain. In these two steps, we can use the utilize the oxygens. Okay, now whenever the deficiency of oxygen is over there, so this in these two steps, TC and ETC, they will be not able to utilize the oxygen. So whenever the oxygen will be less, so less amount of the ATP that will be produced over here. Okay, so within the mitochondria, you can write over here. Let me show with this another color, that as a result we will see the mitochondria, they will be not able to utilize more amount of oxygen because the oxygen is less. So ultimately we will see over here the amount of the ATP that will be decreased, the amount of the ATP that will be less over here. Okay, now whenever the ATP is less, let's talk about the first whatever the problem we will see at the cellular level. So I'll talk about first the whatever the problem we will see in the cell membrane. I'll talk about first the cell membrane aspect. So all of we know that within the cell membrane, let's assume is the cell membrane, the whole cell. Okay, it's the whole cell, and we know within the cells that there are some types of the transporters. One of the most important one, that is the sodium potassium ATPase transporter. Okay, where we see that the sodium molecules are thrown out of the cell, outside of the cell, and the potassium are taken within the cells. So the sodium that will be thrown out and the potassium that will be taken within the cell. But just to conduct this process, just to do the process, we need to use, or there should be utilizations of ATP. Now think one thing, whenever the ATP is less, as a result the cell is not able to throw out the sodium from inside to outside. As a result, we will see the accumulations of sodium, okay, will be within the cell more. So in intracellular sodium concentration, that will be more. Okay, now whenever the intracellular sodium concentration is more, we know that whenever the sodium will be over here, that will be followed by the water molecules. So we'll see there will come what, the water molecules from the outside to inside, because the sodium that will attract the chemo-attracting property, they will attract the water molecules. Now the water molecules will come over here. Now whatever the thing or changes we will see, the first clinical signs or in reversible cell injury, the whatever the thing under microscope we can just see over here, the first changes we will see, there will be what? For example, the cell will be swelled up, just like this. Okay, so we'll see the first clinical manifestations over under the microscope, the cellular or swelling. Okay, swelling of the cell. Let me write it over here. Okay, so swelling. Now whenever the swelling we'll see over here, and then the whatever the next manifestations you can get over here, let me say over those things over here. Okay, now think one thing that for example, whenever the water molecules are just coming from outside to inside, there is chances for the water molecules that can break the plasma membrane. They can break the plasma membrane, for example this part. Now whenever this part will be broken or damaged, whatever the changes we will see, these parts, they will come within the cell and we'll see on top of the specific figure just like this. That is the phospholipids over here, okay? Because we know that this type of the thing, what the cell membrane is made up of the phospholipids. Now this type of the figure we are getting over here, that is known as the myelin figure. Myelin figures. What type of another thing we are getting over here? Myelin figure. I hope you understood. Okay, so first thing I said about what the swelling, cellular swelling we will see over here. Okay, so we can say this is the earliest manifestations of reversible cell injury. Okay, so this is the, you can write it over here, that will be the earliest manifestation. That will be the earliest manifestation. It's not working, so let me show it this one. This is the earliest manifestation. Earliest manifestation of reversible, I'm writing R, cell injury. Reversible cell injury. Okay. Now we can say another term for it, where we are saying the water molecules are coming within the cell. So as a result, we are seeing the cell is going to swell up. Okay. So we can say another name for it, the hydropic change. So another name for it, we can say that the hydropic change. So first we got the cellular swelling, or we got the hydropic change. Second, I already said it, that we can get the myelin figure over here. Okay, and whatever the next problem we will see, for example, whenever the water molecules are coming within the cell, there is a chances the cell membrane, after swelling up, there are some chances for the cell membrane, they will just bulge up just like this. Okay, so by the pressure of the water molecules, we can get bulge just like this. These are known as the bleb formations. Okay, so you can see the bleb formation. Okay, now for example, there is a cell which is having microvilli. For example, there is a cell with microvilli. Okay, whenever we will see the reversible cell injury over here, reversible cell injury, whatever the changes we will see, these blebs, that will be, or these blebs, water molecules will come over here, that will diminish this bleb. So the bleb will be not further seen. Okay, so we'll see the complete cell just like this, because there will be the absence of microvilli. This microvilli, okay, that will be not seen further. Whenever the hydropic changes we will see, or we will see that whenever the cell will try to swell up, as a result we will see the microvilli will come under this figure. Okay, there will be no microvilli in these portions. Okay, the villi will be absent over here. Okay, now whatever the changes we will see, that okay, let me write it over. Myelin figure already said. Whatever the changes we will see in terms of the RER. Now I already talked about the cell membrane. I will talk about RER, rough endoplasmic reticulum. Hope all you know it, that rough endoplasmic reticulum, okay, or simply we can say RER, with that ribosome is getting attached. Okay, so what is the main function for the ribosome? A ribosome that will produce the protein. So ribosome or ribosome, whatever you can say. Okay, now whenever the protein formations will be here, it will have some certain structures, the primary structures. Okay, where the peptide bonds, the primary structures. Then we know that what, there will be secondary structure for it, where we will see the alpha, alpha helix structures, alpha helix, or beta, alpha helix structures. Okay, then we can get the tertiary structures. Then we can get the quaternary structure also. Anyway, now in these cases we know there will be the folding of protein molecules, that is a requirement of the folding. Now in these cases we need to use the word ATP. Here the ATP will be utilized to just have the folds within the protein molecules to have the perfect structure of the protein. Now whenever the protein will be formed in the perfect structure, there should be the ATP utilizations. But I already said you, in case of the hypoxia, less oxygen, less ATP, because mitochondria is not able to utilize the oxygen. So whatever the problem we see in the primary, secondary and tertiary structures of the protein, that will be not formed. Okay, so because of the lacks of the ATP molecules. Now the polyribosomes, the main term over here, that will come over into, that will get breakdown in the monosome. Okay, for examples, is the, let me show it with another color for the ribosomes. See the area here the ribosomes are attached over here, the polyribosomes. Now the polyribosome, that will just come in terms of the what? Monoribosome, okay? So we'll see fragmentations, that will come only to what? In these portions, okay? Now whatever the other changes we will see here, the other changes we will see, the fatty changes. Whatever the fatty changes we can see over here, let me show in this part. So first we will talk about the cell membrane changes, then we will talk about the area rough endoplasmic reticulum changes. Now we'll talk about whatever the fatty changes we can see over here. What is fatty change? Okay. Now, before going to the fatty change, let me tell you that we know within the glycolysis step, whatever the problem we see. So whenever the glycolysis is over here, we know that that will produce, okay, the dihydroxyacetone phosphate. Now whenever there is what, this type of the problem, hypoxia, in these cases we will see this DHAP, they will get converted into triglyceride. Will get converted into triglyceride. Now this triglyceride will go within the liver and they will keep on accumulating within the liver. Okay, for example, this triglyceride, these are the triglycerides. Okay, for example is the now we will see accumulations of the triglyceride, okay, within the liver. Now this type of the problem we will see, okay, there is the fatty change. So the fatty chains we are getting over here, we got over in terms of the what? A rough endoplasmic reticulum. Now we will talk about whatever the changes we can see in terms of the nucleus. Okay. Now let me show it over here. Okay, whatever the changes we will see. Under normal circumstances, we know the nucleus is looking just like this way, where we will see the condensed nucleus over here, the chromatin fiber. This is under normal circumstances. I am writing N. Under the normal circumstances, under the microscope, we will see the nucleus is looking like this. Okay, but whenever we will see the reversible cell injury, we will see there will be just like this problem. That is known as focal clumping. Focal clumping or chromatin clumping. Chromatin clumping we can see over here. So we will see the nuclear changes over here. Okay. We will see over also that what the glycolysis, because okay, let me write it, the fatty changes. We will see the area. Okay, we will see also the cell membrane here. Now another type of the changes we will see, whatever the problem we will see. For example, whenever the oxygen is not able to utilize the, for example, the glucose is coming, we are taking the diet, but whatever the problem you are seeing, that after taking the diet, glucose is not able to oxidize because the lack of oxygen. So whenever the lack of oxygen is over there, so we will see the glucose molecules is not getting utilized. So the body, whatever the changes we will see within the body, that in our body we will see the glycogen is stored within the muscles and liver. Now these glycogens will get breakdown. Whatever the problem we will see, they will get converted into the glucose, no problem here, it will come in terms of the glucose. Now as there is deficiency of oxygen, so there will be no process within the mitochondria. So this glucose, they will get converted into the lactic acid. Okay. You know the lactic dehydrogenase enzymes within the glycolysis, that is not in terms of the aerobic, that is anaerobic. Now whenever the lactic acid is coming over here, as a result we will see the accumulations of lactic acid. As a result we will see the pH that will fall down. So the pH that will drop. Okay. So this type of the problem we will see in terms of the reversible cell injury. The first thing already said in terms of the cell membrane. Then the second thing already talked about what? In terms of the area. Third thing, the fatty changes. And the fourth thing already said about the nucleus. Okay, whatever the problem we will see nucleus. And the last one I said out in terms of the glycogens. So we'll see the pH that will fall down over here. Okay. So this was all about the reversible cell injuries. So if you just watch for the first time, please don't forget to subscribe my channel and press the like. Thank you for watching.